Welcome, Brookings APBIO students, to the third part of our biomolecule slideshow, which is about proteins. Proteins are found in our foods that we eat, and they're very important, provide a variety of functions in our cells. So we're going to look a little bit closer at proteins. They're made from this little building blocks called an amino acid, has a central carbon in the center, has an amino group. We learned about those functional groups in our carbon chapter. A carboxyl group on this end, a hydrogen, and an R group, variable R group, a side chain that could be a variety of things, kind of like when we use X to stand for different numbers in math. The R stands for lots of different groups that could be added to that spot. Notice this is an asymmetric carbon. It's got 4 different things attached to it. So we can have stereoisomers of amino acids. What group is added into that R spot is what's going to give our amino acid and the final protein chain that it ends up in some very unique chemical properties, depending on whether that R group is polar and it likes to be near water, non-polar and very phobic, whether it acts as an acid or a base, can be a charged group. And so that R group is very important in the function of our final protein. We're going to take these little subunits and use that dehydration synthesis reaction that we learned about. We used it to make polysaccharides by joining sugar molecules together, remove the water, and make a bond. And we're going to do that same reaction, only now we're going to be using these little amino acid subunits to join and be our building block, our monomers to build our polymer. Proteins are polymers. So if you look, here's some examples of some different amino acids. Notice they all have a center carbon. We call it the alpha carbon. They all have an amino group. That's where amino acids get their name. A carboxyl group, that's the acid part. This hydrogen can be released into and give off as a hydrogen ion, and that's an acid donating a hydrogen. And each kind of amino acid, notice, has a different group in the R spot. The simplest amino acid is glycine. And what goes in that one for its R group is another hydrogen. This is the only amino acid that is not does not have an asymmetric carbon, because you have 2 groups that are the same here. Glycine does not form stereoisomers, but all of the other amino acids are asymmetric, and we can make isomers because they have that 4 different groups attached to their carbon. Notice the groups can be simple, or they can be long chains, they can have rings, they can be very complicated. There are 20 different amino acids that are used by cells to make proteins. There are a few other amino acids. Think about all the possibilities that we could stick in that spot. But majority of proteins are made by those 20. And make a connection now to our big ideas. Big idea number 1, evolution. The idea that living things share common ancestors, and they've changed over time. They share not only the same genetic code, remember, universal genetic code in all living things, but that code determines the sequence of amino acids and how we build our proteins in a protein chain. And so it's not surprising that if we share common ancestors, that living things would also use those same building blocks to make their proteins. The same 20 amino acids that you use to build your cells are used by bacteria, with a couple modifications, to build bacterial proteins. So if we look at our chain now, we're going to put amino acids together. The backbone comes from that center carbon and joining the carboxyl group of one amino acid to the amino group of the next door neighbor. And it makes a backbone. And what hangs off the backbone now are these side chains, the R groups. So let's see how this is going to work. We're going to make a protein. Here's my amino acid number 1, here's my R group, here's my carboxy group, and here's amino group. We're going to take these 2 amino acids and join them by pulling out a water molecule and making a bond between hooking the 2 amino acids together. And we can make a very long chain doing that. The chain grows in one direction by adding the next amino acid onto the end of the existing chain. And we talked about that before in bio. This is going to happen in the ribosomes. Ribosomes make our proteins for us. And so proteins are polymers of amino acids. If you look at the chain now, this is my backbone determined by that DNA sequence. And the R groups now are going to hang off of that chain kind of like charms on a charm bracelet. Once I get my chain built, the final protein is not just that sequence of amino acids in the chain. The protein comes from folding that chain up into unique 3D shapes. It twists and coils and folds up, and that is what we call a protein. The unfolded form is the polypeptide chain, but once it folds into its 3D shape, we are going to see what we call the final protein. And what's going to determine that shape? It's coming from the R groups. It's coming from those R groups. So we're going to look at a little animation here. The R groups now are going to cause interactions between the amino acids in that chain. And the first thing that happens is some beginning folding. And the two basic shapes that the chain can fold into is it can coil up into a helix, or it can fold back and forth on itself and make what we call a beta pleated sheet. Okay? And then those parts go on to fold and coil even more tightly. So we start with our protein amino acid sequence, folds into various places along that chain, are going to fold into helixes, coil, or fold into those sheets where you connect across between parts of the same chain. And then that continues on to make some more complex folding. Okay? Complex folding, and we get a final 3D shape of our molecule. Some proteins go on even further to take multiple protein chains and join them together to make a bigger, more complicated protein. And so those are the areas, the levels of protein folding that go in to make that final shape of our protein. So we're going to look at what's causing that. What's causing that is the amino acid side chains, those R groups, and where they want to end up. If they're a charged, a polar, they're going to be very philic, and they're going to touch and be near and hang out with water molecules or with other parts of the chain that are polar or charged. They're going to be act like hydrophilic molecules. If it's a non-polar R group, things that have long hydrocarbon chains or rings, those R groups are going to act very hydrophobic. They're going to want to fold so they stay away from water in the cell and end up on the inside of a 3D shape. They're going to fold so they want to be next with other phobic R groups, and so it causes some different folding patterns to happen. Do you need to remember all the amino acids? No, but you should be able to identify that basic shape and look at an R group and say, is that going to be a phobic R group? Is that going to be a charged R group? Is that going to be a philic R group? Basic side chains, some of them actually have a charge. If there's amino groups in your R chain as well, it can cause those to have an actual basic R group and interact, ionic interactions, other kinds of interactions. Remember our sulfhydryl group, we learned about that functional group, has a sulfur in it. There's one amino acid that has that sulfhydryl. Methionine is an amino acid that has a sulfur, but notice it doesn't have the H at the end. It can't make disulfide bridges. This one though, amino acid cysteine, has that sulfhydryl group at the end, and it can form disulfide bridges to help with the folding and the shape, a final 3D shape. So the 4 levels of organization. Primary structure is the sequence. amino acid sequence, and it's determined by the DNA code. So changing that sequence can also change that shape of my protein. For example, we talked in bio about sickle cell anemia and how a mutation in the gene code changes the amino acid in the chain, and that causes some folding changes. This is the molecule of hemoglobin that carries the oxygen for you, and people who have this genetic mutation, their cells, when they get in stressed or in a low oxygen situation, causes that change in shape, that into that sickle shaped blood cell, and causes some medical complications for people who have this mutation, and is affecting the 3D folding of our molecule. Next, we have the secondary structure. This is due to the R groups, phobic and philic interactions. We get the alpha helix coil going. Notice connections between the C double bonded oxygen and one amino acid and an amino NH group in a next door neighbor or nearby somewhere on the chain. We get coiling, or parts of the chains can lay next to other parts of the chain and make what we call a beta pleated sheet. They run parallel. Notice still those interactions between the nitrogen group in one and the carbon group in the next one. Hydrogen bonding attraction between helps to hold those shapes in place. And once I get my helices and my beta pleated sheets, those can go on to fold and coil up into some more complex shapes, also due to the R groups. If you're a phobic R group, you want to be next to or touch or be near another phobic group. If you are a disulfide bridge, have that sulfhydryl group, you can make a bridge, a disulfide connection to another amino acid somewhere else in the chain that has a sulfur group, a sulfhydryl group. We talked about that group when we talked about functional groups. Ionic interactions. If you have charges on your R groups, they can form connections between different amino acids in the chain. And all of those help to fold our molecule up and give it a unique, very characteristic 3D shape. And remember, shape has to do with function. So sulfhydryl groups, making those sulfide bridges, disulfide bridges, are stabilizing. Once we get they can make connections to help hold that 3D shape in place. Notice, here's my amino acid with the sulfur group, sulfhydryl group, cysteine, and when we put them together, we change the name a little bit, cysteine to cysteine. Quaternary structure comes from taking multiple polypeptide chains now that are folded and putting them together into one big macromolecule protein. Examples are collagen in your skin. It's a connective tissue that holds your skins together. Comes from 3 polypeptide chains twisted together. Hemoglobin is another example of a quaternary structure. Has 4 subunits, 4 polypeptide chains, 2 alphas and 2 betas that go together to make the final protein, hemoglobin protein. So look at the 4 levels. Primary structure is the amino acid sequence. It comes from our DNA. What amino acid goes where in the chain? Secondary folding, we get beta pleated sheets and alpha helices, connections between nearby R groups. Hydrogen bonding holds those together. Tertiary structure comes from the R groups interacting with each other, phobic ones touching phobic, philic ones wanting to be next to philic. We have ionic attractions, disulfide bridges to help hold that protein in its final shape. And that's as far as some proteins go. Some proteins only have tertiary structure, but some can go on to make multiple polypeptides joined together, like hemoglobin. We talked about that one. Joining multiple chains would be quaternary structure. Okay, so what do proteins do for us? They're very diverse structurally and functionally. They're involved in lots of different things. If something is doing something in cells, probably a protein. Enzymes, they can be enzymes and help chemical reactions happen. They're structural building blocks. They make cell membranes. They make proteins like keratin, which is found in your hair, your fingernails, collagen in your skin to help structure and hold things together. They can be carriers like hemoglobin, transporting oxygen for you in your blood. Transport proteins, we talked about in cell membranes, the transporters that bring things in and out. Ion channels, hydrogen pumps are membrane proteins. They can be receptors to receive signals, binding to hold cells together. They're important in defense, a part of our immune system. Antibodies are proteins. They help with muscle contraction. The actin and myosin in your muscle cells are proteins. They're important in signaling. Hormone, they can be hormones. Example of a protein hormone is insulin that controls your blood sugar. That's when the people who have diabetes don't make that hormone. They're important in storage. We find proteins stored in some kinds of seeds. If you look on page 78 in your Campbell book, you will see a whole slew of different things that proteins do for you. We're going to look a little closer at one function of proteins, which is to act as enzymes in cells. Enzymes are protein catalysts. Remember, catalysts speed up chemical reactions. And so enzymes are catalysts in living systems that help chemical reactions. And they work by reducing activation energy, the energy that's required to kickstart a reaction and get it going. Enzymes are specific, and they fit like a lock with a key with the substrate molecule they're going to help. And enzymes are not changed by the reaction. They are reusable. They get used over and over and over again. We're going to look at a couple little animations about enzymes and how they work. Why do we need enzymes in the first place? If you look at this reaction without the enzyme present, if I wanted to join these two sugar molecules, they have to line up in the right angle, get the right orientation, get close enough that I can pull out the water to make that dehydration synthesis reaction happen. And they may bounce around for a while. They may bump into each other in the cell, but actually getting them to line up in the correct orientation may take a very long time. If I have an enzyme, remember, enzymes have a special active site where the substrate sits, and it gets those molecules lined up in the correct orientation, close enough together where the reaction can happen. And that's how enzymes help. By decreasing the activation energy, one of the ways we do that is to help orient the molecules and get them in a position so they can interact with each other. Enzymes are specific. Only one kind of substrate is going to fit their active site, and so they only help one kind of reaction happen. They only interact and combine with one substrate. And there's lots of different enzymes in your body that work on different substrates. Every chemical reaction has its own enzyme that helps. So we talk about chemical reactions this year, and we get to photosynthesis and respiration. We talk about chemical reactions. Remember, there's an enzyme somewhere that's helping that reaction to go. Enzymes are reusable. They don't get changed by the reaction. The substrate changes into the products, but the enzyme itself does not change. So once it helps one molecule do its reaction, it can go on to help more and more. They get used over and over again in cells. Protein conformation is important. The shape, that 3D shape, is what's important in making the enzyme, the active site, fit that substrate. And physical conditions can cause enzymes to change their shape. If you have extreme acidity, too much salt concentration can affect enzyme shapes. Temperature, heating an enzyme can cause that 3D shape to change, and that can affect enzyme function. If we change the shape of the enzyme, it can cause denaturing and make it so the enzyme doesn't work anymore. Remember, it depends on that perfect connection between the substrate and the active site on the enzyme. So if I do something like heat my enzyme too high, causes the shape to change. The enzyme, the active site, doesn't fit the substrate anymore, and the enzyme can't work to do its chemical reaction anymore. Okay, let's go back to our slideshow here. So things that can affect enzyme shape, changing the pH, salt concentration, temperature. Some of you may have actually denatured some enzymes this morning for breakfast. We're going to watch a little video clip quick.
An egg provides a good example of how heat can change the structure of proteins. Proteins are found in both the egg white and yolk, and these proteins have different structures in raw versus cooked eggs. Let's consider the proteins in an egg white. In their native uncooked state, the proteins are chains of amino acids that fold into complex shapes. Several types of bonds form between amino acids in a chain, stabilizing the globular structure of the protein. When the egg is cooked, the heat makes the molecules vibrate. The bonds linking the amino acids in chains are strong covalent bonds that withstand the heat. However, most of the bonds that stabilize the three-dimensional structure of the protein are weak bonds, typically hydrogen bonds, which break, allowing the proteins to unravel. When two unfolded proteins make contact, they form new bonds with each other. A large net of interconnected proteins forms, and the egg hardens. Light can no longer penetrate the egg, which is why the clear portion of the egg turns white.
So denaturing our proteins causes them to lose their function, change their shape. It's like leaving your CDs in the back window of your car on a hot sunny day. It causes them to actually warp. And what they're doing is changing their shape. They're unwinding. We call it denaturing. It's one of the reasons why homeostasis is so important in living things. The cells don't like it too much, too hot, too salty, too much acidity, and it's because it affects the function of the enzymes in the cells. When they lose their shape, denature, we say they denature, it causes them to start to unwind, and they may need a little help to get back working again, to get back, and cells have some molecules that help proteins refold if they get denatured. They're called chaperonins. Think of the word chaperone, kind of a guide or a helper to kind of help you along your way. Use your chaperone. Chaperonin, they are proteins themselves, and they're kind of like little containers. They have a lid, and the protein can go inside this container, and it separates it from those extreme conditions that allow the protein to get it refolded back into its original shape, and then release the protein back into the cell. Sometimes if we have too extreme of conditions and it doesn't change, where now we're releasing proteins back into that same condition, we are not going to be able to help the cell get function back. But chaperonins are proteins that you should know about, and they allow they're helpers to help protein guide them back into their correct 3D shape. That's the end of our slideshow on proteins. Stay tuned for nucleic acids.